Hello everyone. Today we will talk about the pluripotency, especially how to induce the pluripotency from the somatic cell. So this is the schematic process of human ES cell line establishment we have learned about in the previous lecture. So in this case, we use the fertilized egg from in vitro fertilization clinic. So isolated egg and sperms are combined and fertilized. Then these fertilized eggs are developed to generate the blastocyst and inner cell mass inside of the blastocyst. And if we crush the blastocyst and isolate the inner cell mass, and then culture those inner cell mass aggregate into the culture dish with feeder cell, then we can observe the colony of proliferating cells which have the uh, pluripotency, which can be proved by diverse way of experiment we have learned. So this is the paper which isolate the human ES cell embryonic stem cell for the first time by James Thompson and Jeffrey Jones in 1998. And this is the first isolated ES cell colony, the portal of first isolated ES cell colonies. So here, there are several characteristics of embryonic stem cells. And this figure shows that the embryonic stem cell established the human embryonic stem cell like H1, H7, H13, H14, compared to the embryonic fibroblast, though they have a much higher activity of their telomerase, which enable the infinite cell proliferation, cell division, and DNA replication. And also these ES cell colonies have abundant expression of cell surface marker, which is characteristics of pluripotent stem cell, such as alkaline phosphate and SSEA series expression. Those are characteristics of the pluripotent stem cell. So we are broadly using these markers to confirm the pluripotency of their established cell lines. And we also discussed this topic in the previous lecture. If we transplant these isolated stem cells into the mouse testicle, we can perform the teratoma assay, and these ES cells form the teratoma, which consists of all the three-germ layer derived tissues, such as gut endoderm derived, and neural epithelium, which is epithelial, the ectoderm derived, cartilage, and bones are mesoderm derived, and those cells are all mixed in the teratoma. And if we found these cells that prove that the embryonic stem cell which are transplanted have pluripotency. So this is the process of the human ES cell establishment. So this is a fertilized egg, the blastocyst derived from the fertilized egg. And if we crush the blastocyst and isolate inner cell mass and plate them into the feeder layer, so these cells are fibroblasts, the feeder cell, which help the stem cells proliferating. And this aggregate of cells eventually form like a colonies, which consist of thousands of cells. And this colony, inside of this colony, these individual cells have a pluripotency, which can be tested by diverse way. And as we talked about before, this inner cell mass derived embryonic stem cell, if we transplant this embryonic stem cell into other blastocyst, host blastocyst, these cells can be combined into the inner cell mass of host blastocyst and eventually generate the chimeric animals. So some of the body part can be derived from this transplanted embryonic stem cell. And that's actually another way to prove the pluripotency of these cells. So we may have a question on a similar way. Can you create the pluripotent stem cell from somatic cells instead of crushing the blastocyst? Because if we crushing the blastocyst, especially in the case of the human, it have lots of ethical issues. Like we consume the blastocyst, and there is a lot of ethical issue such as do we have to consider this blastocyst as a human being? And this maybe if we can create the pluripotent stem cell from somatic cells, such as skin cells or blood cells, easily, that actually remove all the ethical consideration to generate the embryonic stem cell, patient-derived embryonic stem cell, individual-derived embryonic stem cell. So this is the Waddington landscape representation we have learned. So the pluripotent stem cell are differentiating as time goes by to more differentiated status, and eventually they are diversified to various status of cells, and these differentiated cells cannot become more undifferentiated cells in normal status. So this question, can we create pluripotent stem cell from somatic cells, can be translated as, can you erase this existing epigenetic landscape of differentiated cells? Because differentiated cell has very unique pattern of epigenetic landscape, such as DNA methylation or histone modifications, and to make these differentiated cells to the pluripotent stem cell, we have to remove all these established landscape and make it to the initial status. So how to do that? One possible approach would be the somatic cell nuclear transfer, SCNT, and this is actually performed in very early 1960 by John Gordon. And this nuclear transfer, the concept of nuclear transfer is if we transfer the somatic cells into the enucleated egg after removing their own nucleus, if we transplant the somatic cells into those eggs, this egg can be developed normally to blastula and sometimes to the embryonic stem cells, and these embryonic stem cells are pluripotent. Sometimes it can develop the own patches, and in very rare case, such as amphibians, these blastula can be developed into the fertile egg, which means the totipotent. So those cells can become any part of the body, as well including the extra embryonic tissue. An interesting thing is the transplanted somatic nucleus here experience very drastic epigenetic changes. So here, this red line is the epigenetic changes, one of the epigenetic change, important epigenetic change, DNA methylation pattern. And these DNA methylation patterns are increasing during the maturation of oocyte. So inside of the ovary, in female, those oocytes are maturated, and during this maturation, the DNA methylation is highly increased. And the sperm has quite methylated level maintained. So the paternal methylation and maternal methylation is here. But after fertilization, this methylation pattern is dramatically erased and reset, and in modular stage, the de novo methylation is quickly arised in this modular and blastocyst, and the new methylation pattern is established, which is very essential for the proper development of the embryo. And if we transplant the nucleus, nucleus from the somatic cells into the egg, this methylation pattern, because the somatic cells has quite a high level of DNA methylation, right? Which is the signature of certain type of cells like fibroblast or blood cells or muscle cells. And this DNA methylation pattern is quickly erased after the transplantation, um, and the de novo methylation is initiated in the same stage, like between the modular and blastocyst. The de novo methylation pattern is established, increased and established. That means some of the material inside of the fertilized egg make the DNA methylation pattern erased, even after the transplantation of somatic cells. So the important thing is, what are they? What is helpful to erase the epigenetic signature in the transplanted nucleus? Here is the famous experiment performed by John Gordon, which is published in 1962 and eventually got the Nobel Prize. And here is kind of a cloning of the frog by somatic cell nuclear transfer. So frog A, we isolate the egg, and by adding radiation, which destroys the nucleus, we can create the enucleated egg. And from frog B, he isolated the fully differentiated skin cells and isolate the nucleus from the skin cell and fuse these two this skin cell nucleus was transplanted into the enucleated egg. And although the probability is not 100%, but some of the transplanted egg can be developed into the fully mature and fertile frog. So interesting thing is this frog B, cloned frog B, the DNA, the genetic information is exactly 100% same with the frog B, the originated frog B. So it's kind of a clone of frog B. So all the genetic information inside this frog is exactly same with the frog, the original frog. This is a process of the somatic cell nuclear transfer, and let's listen their explanation together.
This video is prepared by my colleagues Dieter Egli and Kevin Egan. And you see here on the left is what's called a holding pipette, which gently sucks on the egg. The egg is surrounded by a membrane called the zona. And you'll watch this pipette on the right first drill a hole into the zona, then go in and suck the nucleus out. And then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. So if we could start the video, please. You'll see now that this drilling pipette is going to drill a little hole into the membrane. You can maybe see a little bit of the hole right here at the next part. This pipette, you'll note, isn't really sharp like a syringe. There you can see a bit of the hole. And now the pipette's going to go in and remove the nucleus. And if you look carefully in the pipette, you'll see a line in the nucleus, which are all the chromosomes lined up. So that nucleus is going to be squirted out now because we don't need it anymore. And there we have an enucleated egg. Now the next step is to take a set of eggs like that, and I'll show you two, and then transfer into them a nucleus from another kind of cell, a fully differentiated somatic cell. So here the enucleated egg is set on the side, that it's held by this holding pipette on the left. There's drilling the little hole in the membrane. Here we go in. Here comes the nucleus from the right. The pipette goes in, and these pipettes are operated with a piezoelectric device. So you can't see it here, but it's like a little jackhammer going very quickly, like a Woody the Woodpecker, getting in there to then squirt the nucleus in. Here we'll see it again. A little hole in the zona is prepared, and now the nucleus is going to be squirted inside. So there's two examples of what's called somatic cell nuclear transfer.
This is very skillful job and need a lot of training, but with some difficulties, we can create the embryonic stem cell-like cells from the somatic cells. So after transplantation, we can create the blastocyst, and by isolating the inner cell mass, we can establish the embryonic stem cell lines, and we can characterize the SCNT-derived ESL by various ways, such as karyotyping or stem cell markers, or differentiation potential, such as teratome formation, and identification of nuclear transfer, such as epigenetic analysis. And this is nice way, so we can create the exactly the pluripotent stem cells, which harboring the exactly same genetic information from the original somatic cells. Although it's known that many animals, such as mouse and ram, sheep, many animals with this SCNT technology, we can create the embryonic stem cell. However, in the case of the primate, especially humans, it turns out it's very difficult and almost impossible to create the embryonic stem cell lines with this way. So it happened 2006, and the Huang scandal try the same way. The Professor Woo-Seok Hwang's group actually used the same way, SCNT, to create the human stem cell line, embryonic stem cell line, and they reported it is possible to create the human embryonic stem cell line by somatic cell nuclear transfer, but it turned out those are fabricated, and the created stem cell line is actually not originated from the donor fibroblast, donor somatic cells. Later, there are a few examples to successfully generate the human pluripotent stem cell from SCNT technology, and they prove that the no similar cloning SCNT is not possible in the human embryo. Here is the details. So we fuse the somatic cells into the not enucleated egg. I mean, they use the ordinary egg, which is not enucleated. So this is the host genome, and this is the somatic cells genome. And after chromosome condensation, they manipulate this embryo with three different ways. So first of all, they remove the somatic cell genome removal, and these somatic cell genome removed egg can be developed at a certain degree with the parsenode. Parsenode is N. The genomic status is N. We call this as a 처녀생식 in Korean. And second case is they didn't do anything to and make it differentiated, developed, and this 3N, because the egg nucleus has N and the somatic cell has 2N, and this 3N embryo, surprisingly, they can develop up to the blastocyst status. And if we generate the oocyte genome removal, this is a traditional somatic cell nuclear transfer, and this 2N embryo, which those 2Ns are all originated from somatic cells, this 2N embryo cannot develop after the after 9 to 12 cell stage, so they cannot differentiate it at all 0 degree to the 0% to the blastocyst. So that's why we cannot create the embryonic stem cell from traditional somatic cell nuclear transfer. So this is the image which have the 3N transfer only embryo, and this GFP4 is expressed in the donor cell origin, and as you see here, this 12 cell stage or modular, this can those all of the cells inside of the embryo are expressed in the donor cell marker, suggesting these embryos are fully originated from donor cell. So we can create the 3N embryonic stem cell. However, the 3N cell does not reflect the original genomic information, which is not very useful for the clinical application or other basic science study, which can be done with pluripotent stem cell. Therefore, the somatic cell nuclear transfer is possible for the other animal species, but it is extremely difficult for the human species to establish the human embryonic stem cell by SCNT technology. So, Shinya Yamanaka consider the totally different strategy. By expression of transcription factor, we may be able to reprogram these somatic cells into the pluripotent stem cell. And this idea is groundbreaking, and it opened up many possible challenges in these days. So with IPS techniques, we can easily create the patient-derived or individual-specific pluripotent stem cell, which is very versatile to study the disease mechanism and finding the therapeutics for the various human diseases. So he got Nobel Prize in 2012. So this is the Nobel lecture of Shinya Yamanaka. And this is very interesting and insightful lecture. So I would highly recommend the full, watch the full version of the lectures. But here I introduce some piece of the important messages from Shinya Yamanaka.
Well, thank you very much, Professor Urban Rendell, for your very kind introduction. It is a great honor for me to be selected as one of the laureates of the Nobel Prize in Physiology or Medicine this year. I am grateful to the Nobel Foundation and Karolinska Institute for having chosen me as one of laureates. Actually, on October 7th, just one day before the announcement, I met the president of Karolinska Institute, Professor Wolfgang Henriksson, in Kyoto. She chaired a session which I attended. When I said goodbye to her after the session, I thought she winked at me. I was not sure. But I'm sure now she did wink at me. So I feel especially honored to share this award with Sergeant Garden because Professor Garden initiated the field of nuclear reprogramming. Without his seminal work done around the year I was born, I could have never be here today. So thank you very much, John, and I respect you a lot for your brilliant head, both inside and outside. So in my talk today, I'd like to talk on 3 topics. First, I'd like to introduce to you my early days in science as a scientist. Then I'd like to discuss about the researches of my own and by others that led to the generation of iPS cells. Finally, I'd like to discuss the potential of iPS cells.
I skipped the lecture a little bit because it's too long. Let's start here.
But Again, when I was working on NAT1, when I was working on NAT1 deficient ES cells, I had to go back to Japan, and I suffered from a disease which I designated PAD. Well, you may wonder what it is, PAD. Actually, I coined the word. PAD stands for Post-America Depression. So, I had such a great time in San Francisco at Gladstone in many ways. After coming back to Japan, I was depressed. I was very badly depressed that I was about to quit from science. But very luckily, two events happened in my life that rescued me from PAD. The first event was the generation of human ES cells by Dr. Jamie Thompson in Wisconsin. Before his success, we only had mouse embryonic stem cells. I was just using mouse embryonic stem cells. I was often told by my colleagues, Shinya, that mouse cells may be interesting, but you should do something more related to human disease, human medicine. So that was one reason for my PhD. But because of his success, it turned out that ES cells are very related to human medicine. Because human ES cells share the two important properties with mouse ES cells. We can expand human ES cells as much as we want, and then we can make various types of human somatic cells from human ES cells, such as dopaminergic neuron, neural stem cells, cardiac cells, and so on. Then we should use these human cells to treat patients suffering from various diseases and injuries, such as Parkinson's disease and spinal cord injuries, and so on. So again, it turned out that ES cells themselves could help patients, not mouse patients, but human patients. So when I first read the science paper by Dr. Jamie Thompson, I got so excited. I still remember that moment. The second event, which rescued me from PAD, was my promotion to NARA. In 1999, I became a principal investigator in NARA Institute of Science and Technology. So I had my own laboratory for the first time when I was 37 years old. As you can see here, this institute has a beautiful campus. They have many good faculty members. They have many good equipments. They have not many, but some technicians who can help me with my mice. And the most importantly, they have many talented graduate students. So because of these two events, I was able to overcome my PhD, and I was able to continue my science. Because I got my own laboratory for the first time, I thought I should make a clear vision or long-term goal of my own laboratory. And I made this as the long-term goal or vision of my lab. I want to make ES-like stem cells, not from embryos, but from somatic cells, from patients' own cells. By doing this, we should be able to overcome the moral, ethical hurdle of human ES cells. Because we have to use human embryos to generate human ES cells, many people are against the usage of human ES cells against the generation of human ES cells and the usage of human ES cells. But if we can make ES-like cells from adult patients' own somatic cells, we can avoid this hurdle, moral hurdle. I thought, at least in theory, we can do this. We can convert adult cells back into the embryonic state because of two scientific findings reported by previous researchers, two scientific streams established by previous researchers. The first scientific stream is, of course, nuclear reprogramming, started by Sir John Gurdon. As you heard from his talk, Sir John Gurdon clearly demonstrated that somatic cells can be reprogrammed back into the embryonic state. The other scientific stream is factor-mediated cell fate conversion, which was initiated by Dr. Harold Weintraub, who showed that a single transcription factor, MyoD, can convert fibroblasts into muscles in mice. So because of these two scientific streams, I thought there should be factors that can reprogram somatic cells back into the embryonic state. By identifying such reprogramming factors, we should be able to make ES-like stem cells directly from somatic cells. However, I had no idea how many factors were required. It could be just one, it could be 10, it could be 100, or even more. So I had no idea how long it would take to identify these reprogramming factors. It could take 10 years, 20 years, 30 years, or even longer. But very luckily, it didn't take that long. It took us 6 years before we identified these 4 factors. In 2006, we were able to report this finding by introducing the combination of only 4 transcription factors, OX34, SOX2, CMYC, and KL4, into mouse skin fibroblasts. We can actually convert somatic cells into ES-like stem cells, which I designated induced pluripotent stem cells, iPS cells. In the following year, 2007, we and others have shown that the same factor combination can make human iPS cells. So I want to use just one slide to show you how we identified those four factors. First, we collected 24 candidate factors with great help of my students and technicians. Among them, these 3 members of my laboratory did an essential work. We also established a simple and sensitive assay system to evaluate these 24 candidate factors. 2 members of my lab, Yoshimi Tokuzawa and Tomoko Itsaka, did this work. Actually, we generated knockout mice and knockout ES cells of a gene which I thought essential in mouse ES cells and in mouse early development. However, nothing happened to those knockout mice and knockout ES cells. Those mice, knockout mice, were just happy. Knockout ES cells were just happy. Only my student, Yoshimi, became unhappy. but it turned out these mice provided us a very useful system, assay system, to evaluate these 24 candidates. So this is another lesson. We should never give up. Then, by using this simple assay system, very sensitive assay system, another member of my lab, Kazu Takahashi, he did a remarkable job to identify the 4 factors out of the 24. So it was indeed these 3 young researchers who generated iPS cells. Yoshimi Tokuzawa and Kazutoshi Takahashi were my very first graduate students. Tomoko Ichisaka is my first technician. Without devoted work done by these 3, we could have never generated iPS cells, at least in my own laboratory. So I'm very, very grateful to these young researchers.
I hope you enjoyed simple and insightful presentation from Dr. Yamanaka. And let me introduce more details about his experiment. As he mentioned, they use very sensitive reporting system, which is the FBX15 knockout mice. Although it makes his graduate students unhappy, this system provides very sensitive identification of the efficiency of reprogramming. Here, the reporter contains the beta-GAO genes. Beta-GAO genes is fusion protein of ROCG and G418 resistance gene. So if we select the colonies with the G418, we can see the evident colony. Only survived colonies express the FBX15, that representing the expression of pluripotency genes. And also we can stain with ROCG staining. We can clearly color these formed colonies with blue color that makes easier. And so Yamanaka selected 24 transcription factors, which is abundantly expressed in the embryonic stem cells, and they used the retrovirus to deliver this transcription factor into the fibroblast. So after the selection, we can see clearly after overexpression of 24 factors, we can clearly see the numerous number of colonies which represent the pluripotent status. And this is the shape of the pluripotent stem cells, which is evidently different from original mouse embryonic fibroblast. And these colonies, number of colonies representing the efficiency of the reprogramming. And they try to minimize the number of factors from 24 factors to 4 factors by removing the transcription factor one by one. And these 4 factors are essential because if we removed one more factors, such as the CMYC, KRF4, and SOX2 and OX34, the number of colonies formed are drastically decreased, that representing these 4 factors simultaneously important and essential for the reprogramming toward pluripotency. And after injection of these cells into the mouse blastocyst, as we mentioned, these injected cells contribute to the certain part of the mouse animal, which is called the chimeric mouse. And occasionally, these transmitted cells are transmitted to the germlines. And in that case, the full adult fertile animal can be generated, derived from induced pluripotent stem cells we injected. So this is the proof of the pluripotency of this 4-factor driven induced pluripotent stem cell. And 1 year later, several groups, including Yamanaka's group, successfully reprogrammed the human fibroblast state instead of the mouse fibroblast with the exactly same factors, OCT4, SOX2, and KRF4, and CMYC. And these fibroblasts can be transformed into the pluripotent stem cells. Pluripotent stem cells, we call this the induced pluripotent stem cell. And it has exactly same genetic information from the human patient, which is very, very versatile and useful to study the human disease and also developing the therapeutics of these human diseases. This is the live imaging of the reprogramming, and after the delivering of 4 factors, and this image, they use the reporter system of the pluripotent gene reporter with the venous fluorescence. So after long-term culturing, you can see the division of cells here, very quickly dividing cells from the colony-like structures, and also they express the venous expression. They express the endogenous pluripotent genes. And these colonies are fastly expanding, and surrounding fibroblasts, you can notify that those fibroblasts has a very different structure, different morphologies, spindle-like morphologies compared to this colony, the cells inside of this colony, and this is quickly amplified. And we call this colony-like cells as pluripotent stem cells. And this is an example of inducing pluripotency from fibroblast, and also other cell types such as blood cells, hematopoietic stem cells, adipose-derived stem cells, or dermal papilla, which is the hair follicle stem cell, pancreatic beta cell, or neural stem cell, melanocyte. Those all different type of cells can be reprogrammed into the stem cell, pluripotent stem cell, by introducing of transcription factor. So the fibroblasts clearly they require the OSKN. For example, some of the cells doesn't require all these 4 factors because they already express the endogenous level. For example, neural stem cells, they endogenously express the SOX2 and c-MYC, so we can omit the SOX2 and c-MYC for the inducing pluripotent stem cell from neural stem cells. And the status, genomic integrity of the induced pluripotent stem cell is quite important for the studying the future, studying the further studies. And this is an example of the karyotyping. The karyotyping is traditional way to mark up the location of the chromosomes to examine the integrity of the chromosomes. And here is the example of the abnormal karyotyped chromosomes. The chromosome number 1 has an abnormality here, and this abnormality can be generated during the induced pluripotent stem cell. So we must select the good colonies with proper karyotype. And this chromosomal aberration is generated with multiple steps during the reprogramming. So embryonic stem cell, this is the process of the embryonic stem cell deviation. And from the blastocyst, the inner cell mass cultured in the dish. And during the long-term culture of the embryonic stem cell, the aberration acquired in culture because the proliferation, quick proliferation of these cells and multiple mitosis is risk for the generation of the chromosomal abnormality. And chromosomal abnormality during the induced pluripotent stem generation is more frequent because the aberration would be already exist in the somatic cells. For example, if we start from the millions of fibroblasts, maybe some of the fibroblasts already have the somatic mutations or chromosomal abnormalities. And if we generate the colonies from this abnormal fibroblasts, it's naturally those abnormal chromosome transmitted to the pluripotent stem cell colonies. And also reprogramming process is very stressful because it erased all the epigenetic memories in the cells. So those aberrations can be introduced during the reprogramming. And also the long-term culture for isolation of the colonies or adaptation of culture, it also have a risk to generate the aberration required in culture. So multiple way, this chromosomal abnormality or genetic mutation is generated. So we need to be very careful to identify the good colonies with very genuine and genomic integrity need to be examined every time we generate the induced pluripotent stem cell. This recurrent chromosomal abnormality is really different from the embryonic stem cell and iPSC. For example, the embryonic stem cell, the chromosome 12 abnormality is more frequent, and induced pluripotent cell, the chromosome 20 abnormality or chromosome 8 abnormality is more frequent. And also, it can be differential. among different way of the delivery method of the transcription factor. For example, episomal delivery of transcription factor shows more increase of chromosome 12 abnormality. However, the viral delivery of the transcription factor increase the risk of the chromosome 20 or 8 abnormalities. And aneuploidy, or the abnormal number of chromosomes, those there are 3 copy of chromosome here in this case. These are also frequent chromosomal alterations during the reprogramming factor, so we need to be careful about it. There are numerous diverse type of delivery of these transcription factor into the somatic cells. So Yamanaka's way, they use the retrovirus infection, and retrovirus infection has some problems because it's inserted into the host genome and it potentially generate mutations or abnormality, especially for the therapeutics, the cell therapy. We need to use these cells for the transplantation therapy for the humans, and this integration of virus genes are very problematic. So many groups developed the various way to deliver the transcription, the Yamanaka factors in safe way. For example, non-virus, the episomal transfection is used to induce the pluripotency, or delivering of the protein or synthetic mRNA or microRNA, even chemicals, small molecules, which change the expression pattern of the somatic cells, which is preferentially induce the pluripotent stem cell status. And probably there are more safer way in the future will be developed with other approaches. So traditionally, somatic cell nuclear transfer, the nucleus of the somatic cells transplanted into the enucleated egg, and it can rapidly generate the pluripotent cells in some species like a mouse, but not in humans. And cell fusion, the pluripotent stem cell, also reprogram the somatic nucleus and induce the pluripotency. And Yamanaka's way, the transcription factor expression, or SKM expression, slowly, it takes more than 10 days. However, it efficiently induces the pluripotency in the stem cells from the somatic cells. And as I mentioned, the small molecules are useful and safe way to induce the pluripotency from the somatic cells. These small molecules change the gene expression pattern of somatic cells, and occasionally these altered gene expression induce the status of pluripotency. It also takes us slow. However, the efficiency of the small molecule exposure mediated reprogramming is very, very low. That's also another problem. So here is the description of diverse delivery of the Yamanaka factors into the fibroblast. So they are integrating genome integrating way, which is not safe for the cell therapy or human disease studies, but the integrating way, the virus, lentivirus, adenovirus is used, excisal vector, which eventually excises the construct from the host genome. That's another way which was developed for the reprogramming method. And non-integrating way is more highlighted in these days. So the DNA, the episomal vector is widely used, and DNA-free method is which use the RNA and modified RNA, especially nowadays the process, the RNA products are more evolved, such as RNA vaccine. So this RNA-mediated reprogramming is quite highlighted in these days. And also small molecule or protein is useful to induce the pluripotency from the stem cells. And this is another description of reprogramming method, the non-integrative way. They goes into the somatic cells, and genomic integrity can be maintained. Integrative way, those viral genes are integrated into the iPSC. That makes several problems. So this is the retrovirus was a popular way in past experiment. However, it has problems of integration. So nowadays, people use non-integrating virus such as Sendai virus, which has very high efficiency, or delivery of mRNA, which make the whole process much safer. between these delivery methods, the efficiency of the reprogramming is different. The mRNA, standard virus, episomal, lentivirus shows all different amount of the efficiency. The successful rate is also different, and the time required for the reprogramming is also different. So we need to choose wisely which type of method to deliver the transcript factor into the fibroblast for the experimental specific purpose. This is summary of the characteristics of a reprogramming method, the efficiency of the pluripotent stem cell colony induction is also different. So you may notice that you need to choose the right way to reprogram your cell for your experiment. So this is example of non-integrating mRNA mediated reprogramming. So they deliver the GFP mRNA together with the OSKM factors. So day one, they use the morphology of the cells as like fibroblasts, and eventually after 10 days, you can see the small colony-like cells are formed. And after 2 weeks, we can clearly see the very big colony formation of the human stem cells. So what does reprogramming factor do during the reprogramming process? So there are important mediators such as KRF4. This I is the mistyping, so KRF4, OCT4, SOX2, and CMYC, and they actually have a positive feedback. So the SOX2 stimulate the expression of OCT4, and also KRF4 stimulate the expression of SOX2, and they boost up their own expression as well. And the CMYC is regulating the upregulation of cell-proliferating mediated genes, and also it contributes to loosening of the chromatin structures. And OCT4 to stimulate the expression of NANOG, and which is essential for the upregulation of embryo stem cell genes or downregulation of differentiation genes. So this feedback mechanism is accelerate the reprogramming process, also turn on the endogenous expression of these pluripotency genes. So although we introduced the exogenous expression of transcript factor, but these transcript factor boost up their own expressions and eventually turn on the endogenous promoter of KRF4, OCT4, SOX2, CMYC, and NANOG. And this is step-by-steps of the reprogramming, cellular reprogramming. And as you may expect that the other stem cell progenitor cell may have the more opening of the stem cell promoting genes, and so their reprogramming process is more simpler than the fully differentiated cells. And there are the steps of the gene expression. For example, in the initial period, the inhibition of the somatic regulators, such as the fibroblast specific genes or muscle specific genes, turned inhibited by the Yamanaka factors, transcription factors, and also all the interminate, the activation of pluripotency loci is increased, and erase of the epigenetic memories, and acquisition of the factor independence. That means the exogenous, even though the exogenous transcription factor expression is turned off, but they can maintain their pluripotency by the expression of endogenous promoters, endogenous genes, and complete reprogramming accompanied with the telomere lengths and X chromosome reactivation and the epigenetic memory erasure, and also the in cellular level, the induction of proliferation and inhibition of senescence and apoptotic pathways or immortalization of cells. Concurrently happened during the timing of the reprogramming. So this is another description, and an interesting thing is during the reprogramming process is exactly reversed by the normal developmental pathways. So in normal development, from blastocyst to the adult animals, there is a pluripotent genes expression is reduced, and the somatic cell genes, the cell type specific differentiation related genes are increased during the development of the embryo, and the telomere length is continuously shortened, and developmental gene expression is boosted up during the embryonic development, and also DNA methylation level and X chromosome inactivation is going on here, and epithelial mesenchymal transition. The embryo, the early embryo cells looks like they have a generally epithelial characteristics, but epithelial cells are loosened, and mesenchymal transition is formed during the development. Reversely, during the reprogramming process, they actually showing the characteristics of the reverse stream of the normal development. So early stage initiation, the mesenchymal-like cells, like somatic cells, mesenchymal-like cells, change it to the epithelial status. They are more tightly adhere together and make a colonies. And X chromosome reactivation, the X chromosome in female case, the X chromosome is inactivated during the development. So only one X chromosome is actively expressed and the other one is suppressed. However, during the reprogramming, the suppressed X chromosome is reactivated, and so the transcription status of X chromosome, both two copy of X chromosome is actively expressing their genes during the after the reprogramming. The DNA methylation pattern is erased, and developmental gene expression is reduced, and telomere is elongated, and the somatic cell gene, the differentiated cell-specific genes are reduced, and pluripotency genes are increased. This is the steps which resemble the reverse stream of the normal development. And this is another description of the step-by-step changes during the reprogramming. So OSKM expression, the endogenous OSKM expression, the OSKM, if we introduce the OSKM factors into the differentiated fibroblast, there is a the transcription wave 1 is kind of a loss of the fibroblast specific marker proteins and gain of embryonic marker proteins increased in early days. And the transcription wave 2, I mean, in many cases, the 99% of cells goes to the transcription wave 1. So, however, there is a very big barrier to turn on the endogenous pluripotency genes. So the transcription wave 2 is related to the turning on the endogenous promoters of the pluripotent genes, and only 1% of cells become genuine induced pluripotent stem cell by overcome these barriers between the transcription wave 1 and transcription wave 2. And histone modification and chromatin remodeling happen much faster than other steps, and the new DNA methylation patterns are established after the histone modifications. So this is a description of the epigenetic barriers. So pluripotent stem cell, when the pluripotent cell differentiate to the mature cells such as neurons, they overcome the epigenetic barriers, which is DNA methylation and histone modifications here. And by this way, the epigenetic barriers are also required to reprogram the cells, like the epigenetic factors, make it efficient to the reprogramming process. So if we change the barrier of the epigenetic status during the reprogramming, probably the reprogramming process is more efficiently happened. To modulate these epigenetic barriers, the scientists developed the various way to change the epigenetic barrier to make the reprogramming process more efficient. For example, the chromatin remodelers, the expression of chromatin remodelers make the reprogramming easier. Histone modification, for example, histone acetyltransferase or expression of histone methyltransferase or demethylation is actually increase the reprogramming efficiency. Although on the other hand, the histone deacetylase expression make the reprogramming process inefficient. And also histone variant, various type of histone variant expression also affect the efficiency of the reprogramming. And the DNA demethylase is very helpful to erase the methylation pattern, current methylation pattern in the somatic cells. And the expression of specific link non-coding RNA or microRNA also boost up the efficiency of the reprogramming. So there are a lot of applications of changing the epigenetic status in the somatic cells to increase the efficiency of the reprogramming. So, and another important aspect is epigenetic memory of reprogrammed iPSC, because the reprogramming process cannot completely erase all the epigenetic information. The concept is the resultant resultant iPSC still have some kind of mark, epigenetic mark, which is not erased during the reprogramming process. And those mark, the epigenetic memory, drive the differentiation more preferentially to the original cell type. For example, if we reprogram the iPSC from neural progenitor cell, the neural progenitor specific markers such as H3K4 trimethylation for the active genome, active genomic locus, and those are still maintained after the reprogramming. And if we differentiate these cells, is more preferentially goes to neurons instead of the muscle pate or hepatocyte pate. So they have a more preferential preference to the certain direction of the differentiation because of the existing epigenetic memory after the reprogramming. So this is one of the concepts, and it also suggests that the reprogramming process is not complete. So we need to select the right reprogramming process and rightly reprogram the colonies after the inducing the pluripotency. And DNA methylation is epigenetic marker of the cellular memory. So the somatic cells have the specific methylation patterns. The pluripotency genes are the promoter or enhancers are methylated, and those expressions are suppressed. Developmental regulators also suppressed. The embryonic stem cell, pluripotency genes are actively transcribed without the methylation, and developmental regulators actively expressed. The somatic cell genes are repressed in the case of the embryonic stem cell, that prevent the differentiation of this embryonic stem cell. Instead of, however, the somatic cell genes, specific genes such as fibroblast specific genes or muscle specific genes or heart specific genes, those genes are actively expressed because of the non-existence of methylation pattern. Methylation pattern is not existing. These promoters and enhancers, and those genes are specifically expressed. Then after the reprogramming factor, they remethylate the somatic genes and demethylate the pluripotent genes. However, this erase of the and reprogramming of the epigenetic pattern is not complete. So although the pluripotent genes are demethylated completely, but certain developmental regulators or somatic genes are slightly methylated, and so they are very easily turned on after the differentiation. That actually make the epigenetic memory for their differentiation patterns after the pluripotency inductions. So the reprogrammed induced pluripotent stem cell is very useful for the versatile application. One of the application is differentiation of these cells to various type of cells to study the cellular processes of human development as well as the human disease pathogenesis. So if the embryonic body is a well-established method to differentiate the cells, so the aggregation of cells make an embryo-like environment for these pluripotent stem cells. And if we add the specific factors such as retinoic acid and insulin, and that promote the differentiation of these stem cells toward the adipocyte, retinoic acid also promote the neuronal differentiation. Macrophage, the macrophage differentiation can be induced by multiple cytokines, which is abundantly expressed in the immune cells, and also smooth muscle cell differentiation or astrocyte, oligodendrocyte. The differentiation protocols are well established, and we can generate diverse cell type of cell types which is existing in our body from the induced pluripotent stem cell. That's very powerful aspect of this technology. So, for example, let's look at the differentiation of induced pluripotent stem cell into cardiomyocyte. So, and these differentiation protocols are mimicking the original pattern of the differentiation inside of the embryo. So, for example, the mouse cardiogenesis, this gradient is formed, and the nodal gradient, a Wnt gradient, affect the induction of the primordial of the cardiac tissue, and these transcription factors such as TBX5 or HEN2 or MyoD is important for the identity formation of the heart cells. And so the protocols of cardiac differentiation in vitro from the embryonic stem cell or pluripotent stem cell, the mimicking this pattern of the pattern of the gradient of growth factors as well as the transcription factor expression. So the FGF gradient is formed and suspension cultures and activating FGF or VGF or stimulation, mimicking the original status of the embryonic cell differentiation. So there are also other modified version of the cardiac cell differentiation, but those try to mimicking the embryonic development, original embryonic development by addition of growth factor sequentially in different day of the culture. So iPSC are very promising tools for drug discovery and analysis of disease. As I said, the patient-derived iPSC, especially this iPSC can be differentiated in vitro to the affected cell type. For example, dopaminergic neuron for the Parkinson's disorder, and diabetics for the beta cell in pancreas for the diabetics, and also cardiac disease for the cardiac muscles. So we can create the affected cell types for the human disorders with this iPS technology, and by using this affected cell type specific the source, we can study the disease mechanism of human disease as well as screening for the therapeutic compound, and especially this it will enable the patient specific treatment, which we call this the disease-specific drug, the patient-specific drug can be developed by this unique properties of induced pluripotent stem cell. And also, this patient-specific iPSC can be used for the useful source for the transplant therapy. And many genetic disorders has mutations in their genome, and especially upon the development of the CRISPR-Cas9 genome editing, we can easily repair the genetic mutation inside of the pluripotent stem cell. And after the repairment of the mutation, which is causing the disease, we can in vitro differentiate to generate the healthy cells for the transplant therapy. And after transplanting these cells, probably contribute the rescue of the disease phenotypes of the human patient. So many type of disease-specific iPSC based treatment or studying is published for the numerous disease. So, for example, spinal muscular atrophy is the cell types are induced from the pluripotent stem cells, and many type of other degenerative or genetic disorders are currently actively studied with the iPSC models. So there are numerous applications of induced pluripotent stem cell technologies. So it's very useful to understand the birth defect, the genetic defect during the development. And there is a possibility of generating patient-specific stem cell lines to study the mechanism of different disease in the laboratory. So in a dish, we can study the human disorder without actual patient. And it can create the model of the drug discovery. So we can test millions of the chemicals, the small molecules into the disease-relevant cell type, and we can test the toxicity of the drug before we applying this drug into the actual patient. And tissue engineering also give a lot of promising. So eventually one of the purpose of the tissue engineering is generating the artificial organs which can be replaced when certain organ is dysfunction in the human patient. And does this alter the debates about the use of human ESL or not? I mean, the human ESL has a lot of controversy, ethical controversy, like destruction of the human embryo, and probably the iPSC technology is one of the good way to bypassing the ethical issues suggested from the embryonic stem cell studies before. So let me summarize today's lecture. So nuclear transplantation, the somatic nuclear transplantation lead to the acquisition of pluripotency of somatic cells. And reprogramming involves radical genome-wide changes in the chromatin structures and DNA methylation. Epigenetic markers drastically changed during the reprogramming. There is a step-by-step changes to the mechanistically we can study and appreciate. And first expression of key transcription factor into somatic cells leads to defined reprogramming to generate the induced pluripotent stem cell. And the patient-derived induced pluripotent stem cell can be used for the elucidation of disease mechanism, drug screening, and transplant therapy, which is very promising in future clinical medicine. I would like to say about the egg of Columbus, because it's a very simple idea, right? Just forcefully expressing the transcription factor into the somatic cell, we can enable the transformation of somatic cells into the pluripotent cell. Probably this simple idea is like a Columbus idea to go to west. We may find a new continent. So after getting this idea, everything looks very easy, right? But before getting actual idea and indeed perform the experiment to find evidence to the ability of this technology, this is not easy. Like, the creative thinking can be criticized. This is not a big deal, but it was big deal before actually everything is happened. So we kind of forget about master transition factors, but by publishing our ideas in 2006 paper, we reactivate that kind of concept. Those concepts are long-term forgotten, but this boost up completely new field of the stem cell biology. I never thought, I think others didn't as well, that a combination of only transcription factor would be enough to reprogram cells, but it did. So we can speculate the expansion of our knowledge, which was developed by induced pluripotent stem cell technology compared to the Columbus expedition. So after 10 years after Columbus expedition, this part of the map was not exist in Columbus before the Columbus expedition, but it boosted up the voyage toward the west side, and it the European people know much more knowledge about the America and South America and many part of the other area of the earth. So similarly, the iPSC technology open up the new avenue, and you may see a lot of new things, I mean, which was completely not exist before the invention of the iPSC techniques. So this is the thesis celebrating the 10 years of iPSC. It's already more than 15 years after the developing of the iPSC technologies, and it's really quickly increasing. The field is quickly increasing, and the actual application of these technologies is increasing. We cannot expect how far this technology will go in the future. So I hope you can appreciate the concept of induced pluripotent stem cell reprogramming and the future application of this technology. So I think you can be one who can contribute significantly on the development and future challenges to overcome the future challenges of this technology. So I hope you have more interest in the induced pluripotent stem cell biology in the future. Thank you very much for listening.